I want to give you a really broad strokes view of a field that was really a frontier when I was a graduate student. As Constantine said, my foundation is in butterfly biology, population genetics and evolution. And despite the fact that butterflies are a hyper-diverse group of organisms, when I was a student, there was a kind of a lull in new techniques and questions being asked in butterfly biology. So I was really interested in doing something novel. I wanted to get my PhD in an area that was kind of an unknown. And one of the first things I did as a student was to do a big library project on the extent that chemistry mediates pollination. And the answer was really short, very, very few studies. And I realized if I could coherently learn how to study plant volatiles and link them with pollinator behavior and olfaction and pollination ecology and genetics, I could actually have a pretty decent career exploring and discovering new things. And that's exactly what's happened 25 years later. So for the students out there, it is worth it, that extra library project, because it might show you, in the long term where you could find a real opening in science. And science is really about discovering the unknown. So here's a structure of today's talk. It's going to be, as I said, really broad strokes. I want to talk a little bit about plant-pollinator networks because the social network approach to pollination has kind of swept the world of pollination by storm, particularly in agricultural settings. And I want to ask a little bit about what the chemical aspects of flowers have to do with that landscape view of the world of pollination. Then I want to talk about direct and indirect selection. One of the many revolutions going on in biology at the moment is phenotypic selection studies, the chance to take somewhat sophisticated regression models and ask what constellation of traits or interactions between traits is associated with seed fitness. And so I want to talk a little bit about what we're learning about scent in that regard. Geographic variation and interactions between plants and pollinators. This is the stuff of evolution. Because chemical ecology is so founded on chemistry, the whole concept of variation has been understudied in plant volatiles. And so a lot of my career has been spent trying to backfill the sense of what's known within a population, between populations, across the geographic distribution of any plant. There's got to be variation in scent. There's variation in everything else we can measure. And so then the question is, well, what does it mean ecologically, evolutionarily, physiologically, et cetera? And I want to end with by talking about reproductive isolation. For evolutionary biologists, this is the coin of the realm. And the literature, again, like many of these other topics, is remarkably devoid of floral chemistry. So I want to talk about a few very famous systems in which chemistry appears to matter in terms of providing pollinator-mediated reproductive isolation. I have mimicry and deception at the bottom. This is something that people on campus know that I do. When Paul Cooper invited us to work with the Titan arum several times in the last few years, his wonderful plants, that's one of the greatest celebrations of floral deception on the planet. I've given several talks about that. I won't really touch on that today, just to mention that it's a big part of maybe an overrepresented aspect of floral chemistry, that because deceptive plants are so strange, and in some cases so captivating of the public's attention, we have a fairly good understanding of what makes them stink and what makes them work. All right, so I'll begin where I began as a student. I was really interested in convergent evolution, and it's been well known for literally centuries that convergent evolution across the planet often results in suites of traits that are largely associated with specific kinds of pollinators. What I've given you here is a snapshot of two guilds. This is the South African guild, 30 degrees south latitude in the grasslands of the Indian Ocean coast of South Africa. And this is 30 degrees south latitude on the other side of the planet in the grasslands leading up to the mountains of the Sierra Chica of Cordoba, Argentina. And what you see here are two guilds of night-blooming plants that have converged on a tubular morphology, putting the nectar at the bottom of the tube, making a hawk moth put its tongue all the way in, getting the pollen on the body of the moth as it drinks. and smelling like essentially Easter lilies or jasmine or gardenias. This is the source of a lot of natural perfumes, and chemically, they have a fingerprint. They converge on this sort of loose pattern of acyclic monoterpene and sesquiterpene alcohols, indole and other nitrogenous compounds, and then these aromatic ring compounds with alcohols and esters. They smell sweet to us, probably because of the absence of things that smell nauseating. Indole, I think you could argue, is not necessarily a pleasant compound to most people, but at low concentrations it does smell like jasmine. Anyway, this is the kind of thing I was looking for as a student. Chemistry that I could smell, that was easy to analyze using gas chromatography and mass spectrometry, and that might represent the outcome of selective forces. In this case, selection across the planet to attract large nocturnal hawk moths as pollinators. But one of the great biases in chemical ecology, and especially in the study of floral scent, is that it's all about pollinator attraction. When you analyze the floral headspace of a night blooming plant like this tobacco, you find some remarkable things. So what I've done for you here is to show you the chromatogram that you get when you collect scent during the first hour of anthesis of one of these night blooming flowers. Okay, and I've color-coded the different classes of compounds that you might find being emitted by this flower. Ecologically and functionally, what do they represent? Okay, so these compounds here have been shown to be pollinator attractants. Okay, same is true for some of these as well. These plants tend to have redundant blends of things that are attracted to pollinators. But there's also plant defense compounds in here. There's compounds that are trapped in the floral headspace but have nothing to do with pollinator attraction. They have a lot to do with protecting that nectar, keeping thrips and nectar robbers and other creatures from hijacking the reward that is really intended for pollinators. But there's more. There's phylogenetic signal here. All tobaccos make this group of compounds here. At least all tobaccos in this sort of Argentinian and Brazilian clade produce 1,8-cineol, the eucalyptus oil, and 7 other monoterpenes that are biosynthetically related to it and made by the same enzyme. So that's a shared derived trait. That's a synapomorphy, okay, for phylogeneticists. Independent of how these are pollinated, whether they're self-pollinated or pollinated by hummingbirds or hawk moths or whatever, they all do it. And then the biosynthetic part is that some of these compounds simply can't be made as individual products of structural gene products of enzymes. That if you make this alcohol, you make the ester that's associated with it. If you make these aldoximes, you make the nitriles too. That isoleucine and leucine are simply going to give you suites of volatile compounds that are structurally related. So I should probably not use the word noise, because everything in chemical ecology has multiple functions. You just have to be creative enough and open-minded enough to do the right bioassays, whether they're antimicrobial, whether they're modulating pollen tube growth, whether they're changing the pH of a part of the flower. We're limited in our creativity. We often don't know functions because we don't ask in appropriate kinds of assays. So any blend of volatiles from a flower will give you this mix of function and origin and questions. Okay? And that's one of the things I love about my field, is that there's still a lot of decoding to do, just from the plant end, not even from the animal end. Okay, and the caveat in the corner is phenotypic plasticity, okay? That as so many of you know very well from your own work, plants are not static. Plants respond to stress, to soil differences, to being attacked and induced by enemies, to fighting off pathogens, to pre versus post-pollination processes, et cetera. There's a lot of plasticity in here, and then that is also a huge open area of current endeavor. All right, so let's talk about pollinator networks for a moment. It is not unusual these days to open a paper in a high-impact journal and look at some new modular analysis of a plant-pollinator social network, on an island where they're endangered, or in a grassland meadow in Germany that's been glaciated, or in the rainforest canopy of Peru. This is kind of a growth industry. And typically, the points that people try to make are that you have this kind of structure in webs. So you have a generalized pollinator like a bumblebee that visits everything. Okay, the numbers here are insects or pollinators. The letters are flowering plants in a community. And so, wild carrot gets visited by essentially everything. So these guys have high connectance, right? They're talking to everybody in their community, essentially. And if this flower blooms for a very long time, it might be feeding or subsidizing everybody in that community. So that's something, that's a really interesting idea to come out of network studies. What's often also interesting is that the obligate specialists, figs and fig wasps, get stuck in the corner. And there is a tendency in this literature to say they don't matter, okay? Like, let's focus on these guys because the rest of the community depends on that. So for instance, if F were a lady slipper orchid, okay, and they only are pollinated by queen bumblebees, then you need wild carrot to have lady slippers because you've got to be feeding those bumblebees for the 51 other weeks of the year when the lady slippers aren't blooming, okay? That's one of the great take-home messages of these studies. This is a bad one, okay? Because if you invert this web, the fig fruit are feeding everyone, okay? In a rainforest, anywhere on the planet, that's the hub, not that, okay? So ecology is more complicated than these webs. These webs are not static. The problem is that most people who work with these webs basically model them in Brownian motion. You know, they look at density dependence in a forest or in a field and say, okay, if that's the most abundant thing, then everything should be visiting that just because they're following optimal foraging rules. But insects don't necessarily work that way. They have nervous systems that might be repelled by the odor of Daucus carota, okay, in which case they won't visit it even if they should. Or they might learn, you know, I shouldn't go there because I'm a hummingbird and I can't get anything good out of that, even if it's the most abundant flower in the habitat. So there's a lot of oversimplification that goes on. And the interesting part that I want to show you is that these are places where literatures don't merge. So here is an example of a hub. Andrena nigroenea is a southern European bee. It's a very abundant one. And this is a kind of simplification of a network from Greece, studied by our colleague and friend Theodora Patanidou. Beautiful work, terrific ecologist. So here's Andrena visiting all these different European plants, pollinating some of them, taking pollen and nectar from all of them, okay? And that's all been modeled as, you know, density-dependent ecology. Here's the male of that bee species pollinating Ophrys orchids, same habitat, okay? And that's chemistry, right? That's sex pheromone mimicry. That male is not looking for food, it's looking for reproductive access to females. And so, you know, the rhetorical question that I always have when I read these papers is, why would there be turn-on chemistry for this and like go density-dependent for that? Like, why is that the only chemical link in this network? So what I'm going to show you next is a kind of summary, quick summary of a study that I did with colleagues in Europe asking that question, okay? So here we looked at two major hubs in a network, two flowers that are really abundant and bloom everywhere in a meadow and get visited by everything, more or less. Okay, so Achillea millifolia, the yarrow, and Cirsium arvensi, the Canada thistle. So these are not specialized plants. They are invasive. We have them here. We can do the study here if you want. And yet, they're not super generalized because here's their network. Okay, Canada thistle is visited by honeybees and bumblebees and some flies and some solitary bees. But yarrow is more likely visited by syrphid flies and muscid flies and some beetles. Okay, so what we did is we took pentane extracts of these two species and we sprayed them on each other. We basically temporarily swapped their scent without changing anything else about their density, their height above the ground, their color, or their morphology. Those are all the traits that pollination biologists think are driving these interactions. And what we got was this gorgeous 15-minute temporary spot where while the extract was still there and being emitted from these flowers, we switched around all the linkages in their little network. Okay, so insects that were going to Achillea formerly were now going to arvensi that had the Achillea, you know, scent on it, and vice versa. We basically nullified the network. And at 5-minute intervals, we watched it decay all the way back to the starting. So that worked out better than we hoped. We thought, you know, we were worried that the pentane might simply kill all the insects or that there might be some strange, you know, novelty attraction to pentane because foraging insects are often interested in anything volatile, for example. But instead what we got was this transient modification of the networks based purely on scent. Once we watched the animals, once they landed, they realized relatively quickly that they were not where they intended to be. And so they didn't continue probing at the flowers. There are other aspects of flower learning that aren't driven by headspace compounds. But it was so fascinating to us that the currency of this kind of study, which is the link, was completely shifted simply by swapping scent. Okay, and then the follow-ups. Here's how the chemistry differs. Okay, yarrow is all about terpenes, mostly sesquiterpenes, and Canada thistle is all about aromatic compounds. So chemically, they're really different, even though they're both asters. And that's not saying much, right? Because 20,000 species of asters. But you can see that swapping them really changed their scent in a major way. These are my co-authors, Robert Juncker and Anna LaRue. This is part of her thesis in Salzburg. And what you see here is that when you do the bioassays, there are different reasons for these animals to have shifted. In one case, there's repellence. So the odor of yarrow actually repels honeybees, whereas they are attracted to the odor of the Canada thistle. And we repeated this with syrphid flies, with bumblebees. It's all in the publication. But in essence, through different mechanisms, repellence, attraction, masking, et cetera, the common theme was that the odor of these flowers really mattered in terms of this taxonomic diversity of insects that were visiting them. So if one of the major criticisms of the study of floral scent is that it's specialized and trivial to special orchids or whatever, this is the kind of study that's a good rebuttal to that, because it's about generalized trashy weeds that are visited by the kind of pollinators that most people study. Okay. All right, direct and indirect selection. Let's talk about this a little bit. This was a study done here by an EEB student, Amy Peraknowitz, who's now working in Uppsala, Sweden. Amy studied a nondescript local wildflower, Penstemon digitalis. It's variable for anthocyanin flush in the nectar tube. It's been studied before by people in the Midwest. There's a nice little literature on its pollination biology. It's generally pollinated by bees. So what she did is she did this kind of untargeted study where she measured lots of traits: color of the flowers, flower size, daily display, the number of flowers per plant, height above the ground, and then she collected floral volatiles and included them in a big statistical mash, okay, being regressed multiply with controls for multiple comparisons against seed fitness. Okay, this is typically done, kind of Landy Arnold approach to evolutionary correlation studies, right? And what she found was that color, which everybody thinks is so important, in this case didn't matter at all. And flower size didn't matter at all as well, not significantly associated with seed fitness. Daily display, number of flowers, and total floral scent did. So of course, like any statistical approach, when you get that kind of result, you're allowed, you sort of justify going in and searching for the target of selection, right? So what she found was that of all the scent compounds that were released by this flower, the only one that had significant selection differentials and selection gradients was linalool. Okay, so linalool is a really common monoterpene alcohol, smells like Earl Grey tea. Okay, it's in bergamot oil. It's a compound I worked on as a graduate student, looking at its genetics. And so that's great. You know, of all these compounds, what's linalool doing? It was not a major peak in the headspace. It was a tiny little pimple on the chromatogram. So what we did was a kind of SPEME dissection of the flower. We collected and dissected a lot of flowers and looked at where in the flower are these compounds being made. So I'm just going to walk you through a dissection. This is really fun to do. Here's the flower itself, and we sort of controlled for flower mass and put them all in scintillation vials and let them equilibrate. And so this nectar spur back here, less than 10% of the mass of the flower, that's where the nectar is secreted, that's producing all the terpene chemistry, all of it. Ocimene, the sesquiterpene hydrocarbons, and this little tiny peak of linalool. That's the target of selection. It's this tiny little thing. You can see the glandular trichomes on the adaxial surface of the corolla tube. These are putting out mushroom oils, mushroom alcohols, right? These are classic allelopathic compounds that between fungi. They inhibit germination of spores. It would be a great way to protect your flower from not accumulating any yeasts or epifloral fungi, but really different chemistry. The pollen, the anthers themselves, completely different. Okay, they have this long aldehyde here, and then this pyridine. Pollen, of course, is if it's being collected by the bees, they would be very conscious and aware of pollen chemistry, and pollen chemistry often is quite distinctive. But here's the killer. This little peak here, present on the staminode. All penstemons have four fertile stamens and a sterile staminode. The staminode is usually to kind of position the bee and push them up into the stigmatic surface, right? They have this sort of clam-like stigma. The staminode here smells of cinnamon red hots. Okay, this is methyl cinnamate. And so imagine this is the first thing the bee touches when it visits a flower, and it immediately gets this like really striking phenylpropanoid odor in its face. Wow, okay? And yet there's several studies of this plant that don't even consider that any chemistry might be happening here. When the bee inserts its entire body like a glove, like a finger in a glove, into this flower, and it's got all this chemistry coming at it, I want to leave you with this idea of a disconnect. This field is a huge disconnect, okay? But the interesting thing was there's the linalool in the nectary, in the nectar tube. It's a small peak. Somehow that is really speaking to the bee. And the question is how, okay? What we found, I won't show you the slide, it's one enantiomer of linalool, because linalool comes in two flavors, okay? And it's present in the nectary, it's actually polar. So it dissolves in nectar. So one of our questions is it a flavor rather than a scent? And is it something that the bees learn by taste and then associate with the presence of nectar? Because it's in the nectary, that would be a good thing for it to learn. But it's also antimicrobial against yeast, and it could simply be protecting the nectar against yeast, and it might be that you get more seeds produced when you have more linalool because the yeasts aren't making the flower sick. So these are hypotheses on different levels of organization and analysis, and they're all reasonable. They're not true alternatives, okay? And because these compounds are multifunctional, it's a mistake to think that only one can be true. So there's more work going on in that system, but I wanted to share it with you because it was not only unexpected by the people who study penstemons, who never talk about scent, but if you were just to take a kind of pluralistic look at chemistry here, you wouldn't pick linalool as the one that mattered. Okay, you'd get there after 12 other compounds because of the relative abundance. All right, let me give you one more example of this kind of a study. This is work that I did with Candace Galen, and I was so happy to do this because she's one of my heroes as a scientist. She worked on these crazy plants, Polemonium viscosum. They're wildflowers in the Rocky Mountains. They have a large distribution, and they're famous because she made them famous by studying their altitudinal transects, that in the Krummholz, okay, at the tree line, they've got these narrow little flowers that are pollinated largely by syrphid flies, and they smell extremely skunky. So the trichomes are probably making thiols, okay? They're very pungent. They would fill this room if I brought them in. Above timberline, you don't get that. So she described it as a kind of altitudinal polymorphism of sweet and skunky. It's not quite accurate. The polymorphism is the presence of skunky odors coming off the trichomes and the calyx. Above timberline, that doesn't happen. But the corollas always smell sweet. And they always smell sweet because they're making 2-phenylethanol. Okay, so that's kind of like a hyacinth odor. You know, 2-phenylethanol is everywhere. It's in yeast. It's in a lot of sweet-smelling flowers. It's a very penetrating, sweet compound. But it's also, as you'll notice, polar alcohol, and it gets into the nectar. So the really interesting thing here is that for 30 years, Candy showed that flower morphology, the kind of flare of the nectar tube, was associated with efficient pollination by, in this case, flies, in this case, bumblebees. The wider the flare, the easier it is for a queen bumblebee to get into it. Okay? The problem is that the wider the flare, the easier it is for an ant to get in there. And at that elevation, in that severe habitat, ants are really sugar limited. So these formica ants are formidable flower predators. What they do is they go into the flower and they actually clip the style. They snip the style, so they female neuter the flowers. There's no female fitness, there's no seed fitness in a flower that's been visited by an ant. So she has always thought of corolla flare as an expression of trade-off between effective pollination by bees and protection against invasion by ants. Okay, and she's mathematically modeled this. She's looked at exclusion experiments where she's kept one or the other out and looked at what happens in the progeny, et cetera. Elegant studies. What we found was that there was this strange relationship between corolla flare and the concentration of 2-phenylethanol, or the dosage emitted by the flower. The more narrow the flowers are, the less scent they make. The more flared they are, the more scent they make. And so we did some swapping experiments where we washed out the flowers and put our own solutions of sucrose with 2-phenylethanol into it, with different concentrations. And we did bioassays in Petri dishes and in choice assays in tents. And what we found was that these ants are killed by 2-phenylethanol. They're strongly repelled by it, and if they can't leave the flower, they'll die. So it's toxic, it's deadly toxic to them at the concentrations that the flowers make. It's amazing. What's even stranger is that the bumblebees also are repelled by the highest concentrations of 2-phenylethanol. So what that means in the end is that the flowers with the wide corolla flare can afford to have wide corolla flares because they have a lot of 2-phenylethanol. They keep the ants out, and they get pollinated effectively by bees. But the fact that the bees are repelled by it means that they'll only visit 1 or 2 flowers per plant and then move on. So it's really enhancing outcrossing for these plants. This is not something that was modeled in the literature. We thought there might be balancing selection between bees attracted to the same odors that ants were and the plant having to kind of balance, mitigate their damage by the ants. But in fact, 2-phenylethanol repels everyone. And that's fascinating. We did not see that coming. Okay, geographic variation. I want to talk about California for a moment, because this is a great place to study geographic variation. This is a plant, Lynanthus dichotomous. It's in the phlox family, that was studied by Gretchen Laboon, who was here this fall on sabbatical, and her master's student, Sally Chess, working with me in South Carolina. There are 2 subspecies of this plant, one north of San Francisco Bay and one south of San Francisco Bay. They bloom at night. They smell like jasmine. They don't have a drop of nectar in them. So they're basically cheating moths by tricking them with white reflection and fragrance. Okay? And what you see here is that the northern subspecies actually opens at midday and stays open at night. And that's typical because Northern California is colder and wetter, and many plants tend to be pollinator limited in Northern California. And so this is sort of has been interpreted as a reproductive assurance strategy where the plant is opening itself up to more possibilities to visitors. We grew 20 plants from each of these 5 north and 5 southerly accessions in a common greenhouse, randomized their positions. Okay, so we took away the geographic and environmental differences of Northern and Southern California and asked, how different are they? In terms of emission rates, they're not different at all. But in terms of chemistry, they are. So the blue triangles are the southern subspecies that bloom only at night. And they produce these lilac compounds that are sort of global attractants for noctuid moths. It's exactly what you would expect them to do. The ones that are the green ones are the northern populations. So they're shifted a little north in this multidimensional scaling analysis here. But they're shifted because their chemistry is different. They're adding these generic aldehydes, benzaldehyde and phenylacetaldehyde. They attract noctuid moths too, but they also attract everything else, bees, butterflies, syrphid flies, et cetera. So what's actually happening here is the northern subspecies is opening during the day and basically saying, come hither, we'll take anyone, okay? Whereas the southern one is saying, I'll take my chances with the moths. We're in a good place for moths. We'll open at night, we'll close in the day, and that's going to be the way we do things. The trade-off being that if you have a more generalized system, your pollen might not go to your own species, right? You have a higher risk of what we call heterospecific pollen transfer. So that's often the trade-off in mixed mating systems and in pollinator specialization. That if you're pollinator limited, you may have to deal with heterospecific pollen transfer. Okay, so same habitat, what else is going on? Moths, let's talk about moths. There is a continuum out there in moth pollination, from nectar-based pollination where moths are acting like bees or hummingbirds, to nursery pollination like yuccas and yucca moths, where there's no nectar or pollen involved. The moths are coming to the flowers to use them as hosts. Okay? And then in between, is this wonderful system, Lithophragma in the saxifrage family, pollinated by these Graea moths. I'm going to spend a few minutes talking about them. So John Thompson is my colleague and collaborator on this. John is best known for his work on the geographic mosaic theory of coevolution, the idea that there are hot spots and cold spots across the range of a plant and its pollinators. So the Lithophragma flowers, these moths are so docile that you can cut the flower in half and film them and photograph them actually doing the job, visiting the flowers, either laying eggs in them or drinking nectar from them. They're unperturbed. It's remarkable. So in parts of their range, they're pollinated by this moth that uses it like a yucca moth, lays eggs in it. In parts of their range, they're visited by this moth, which lays eggs in the stems, but visits the flowers for nectar, not as hard on the seeds. And in some of their range, they're visited by bees and bee flies and are pollinated like a lot of other flowers. Okay? And so there's benefits to these kinds of things. You get high seed set and frequent visitation here. The costs are, again, low constancy and patchy distribution of these guys. Here you have an obligate animal. He's always going to be there for you, but you're going to have to pay in seeds. If you're going to get pollinated by this animal, you have to give up some of your reproductive success. So what John's done is, across the West, he's looked at these different populations and asked, where are the places where the plant is stuck with the pollinator that lays eggs in it? Okay, where it's got to tolerate that animal somehow, and there should be really strong selective pressures, and those are hot spots, okay, of reciprocal effects on fitness. Then there are other places that are so-called cold spots. If you've got a co-pollinator, maybe you can fight back against this guy if you know you're gonna, if you have a reliable pollinator that isn't costing you ovules or seeds. There might be a different selective regime in a place like that. Or in a place like this, where there's 3 co-blooming species of Lithophragma, you could play a little bit of a shell game where some years the moths will go to your neighbor and not to you, and the selective pressures might be diffuse there. Or if you have the one that's nectaring instead of the one that's laying eggs, that might shift around the selective pressures somewhat. So this is a marvelous system, and it's not at all limited to Lithophragma. This happens in salamanders and garter snakes. There's many classic studies of geographic mosaics. But this is a good one because the seeds don't go very far and the pollinators don't go very far. So there's not a lot of gene flow and there's a lot of local adaptation. So what we asked was, could we actually see a footprint of those different selective pressures on chemistry for these flowers? Knowing that, again, we could collect bulbils in the field, grow them in a common greenhouse, take away those environmental differences and ask, how different is their chemistry? So what we found, there are 3 species here, Lithophragma parviflorum from up here in the Palouse, Lithophragma bolanderi from down in Santa Barbara, et cetera, and Cymbalaria from the eastern Sierra. The color coding of the pie charts is simply a way to show you how different the chemistry is. Each color is a different class of volatile. These flowers are as diverse, and these are barely species. Their differences in chemistry are on the level of families of other angiosperms. So there's something really funky going on in terms of generating chemical diversity in this lineage of plants. And this is just, again, an ordination to show you how different the chemistry is in sort of 2-dimensional sense space. But we did 2 experiments. One was a Y-tube binary choice experiment, and it's kind of 3 by 3. We had moths from 3 populations, and we gave them a chance to interact with each other's host plants, okay? And the hypothesis being, if they're showing local phylopatry, they should only respond, or they should preferentially respond to their host plant from their population. And that's what you see here. The dark bars show you statistically significant preference in a Y-tube of a moth choosing the flower from its population by scent. No visual display, no other cues. And that always happened, okay? Sedgwick chooses Sedgwick, Turnbull chooses Turnbull, Marble chooses Marble. Not surprising, but satisfying, okay? When you then take those female moths and put them in a no-choice assay with one of those 3 plants and ask them, how many eggs do you lay? Again, this is what they do. They lay the most eggs on the populations that they come from. Okay? So there's very strong local phylopatry here in terms of females choosing their hosts, and it appears to be driven largely, if not entirely, by the floral scent chemistry. Okay? And here's a sort of image of the thing of what's to come. These are each pie is a population that we've studied. Okay? And it's a lineage of 8 species. They're very, very closely related. There's 2 independent origins of pollination by these moths. And if you look at the chemistry here, and each color is a different class, a different biosynthetic pathway that's being invoked, you see some really astonishing things. The brown ones are all populations that have bees, okay? And they're shifting to the odor of tea roses, okay? Which is a universal attractant for andrenid bees. So that's really cool. There's a lot of detail in here. Obviously, we're missing all of Oregon, right? All of the Great Basin. There's so much left to do in this system. And yet, I can't cite another system in the literature that's this diverse and complex at such a small phylogenetic scale. Okay, how are we on time? When do we usually end?
7 minutes.
7 minutes? Okay, good, we're almost done. I'm gonna finish then by talking about reproductive isolation mediated by scent chemistry, and then we can open it up to questions. The best studied cases, beginning with Fern Grant, of ethological isolation have to do with columbines. Okay, so this is a recent paper by Scott Hodges and his students showing the direction of evolution from bee pollination to hummingbird pollination to moth pollination that involves color and orientation and spur length. And Hodges and et al. Have nicely shown over the years that if you reorient the flowers, the moths can't visit them. If you reorient the colorful ones, the moths still don't like those. So there's obviously aspects of posture and color that matter to what a moth chooses in the Sierras to visit. So that's what's called ethological isolation, right? An animal whose behavior is mediating pollen flow, okay? Whereas mechanical isolation has more to do with, is there a morphological aspect, not scent or color or posture, but something about the fit between the animal and the flower that determines the efficacy of pollen transfer. And here, this is all about spur length, right? That the number of pollen grains that's transferred increases as the spur length is experimentally lengthened to infinity. Whereas the number of visits don't matter. The moth doesn't care how long the spurs are. But if it has to reach further into the flower to get to nectar, it's going to get better physical contact with the flower. So these are classic studies. These have been known for a long time, and they never include any mention of scent at all. Monkey flowers, same thing, just a paper last year on scent with monkey flowers, but typically these things are assumed to be sort of color and density driven as always. Okay, Ipomopsis is another group of plants, phlox family, that Vern Grant found really interesting. He wrote a beautiful book in the 60s that kind of ignited the field of pollination syndromes. I spent much of his career studying them. As you can see, there's several transitions in the Ipomopsis lineage between hawk moth pollination and hummingbird pollination. So in the same places as columbines live, using the same animals. So if you took Ipomopsis, columbines, and monkey flowers and mapped them over the American West, essentially, 2 genera of hummingbirds and 1 genus of hawk moths are driving all that. Vern Grant's whole career, essentially. So we wanted to look at this, and I did it with Diane Campbell, who spent much of her career studying reproductive isolation between these 2 species of Ipomopsis in western Colorado. So at low elevation near the Black Canyon of the Gunnison, they are completely reproductively isolated. All right, so here's aggregata, the red one, is visited only by hummingbirds. Here's tenua tuba, the white one, visited only by hawk moths, and never the twain shall meet. There's no introgression. These things are completely isolated. The nectar is not present at the right time for the different animals to visit, et cetera. If you go up the ridge, 1,000 meters, sorry, 3,000 feet, what you get is something different, okay? It's cold at night. Hummingbirds still go to red flowers, hawk moths still go to white flowers, but they start to blend. The hawk moths start to forage diurnally. Okay? We don't know if it's because it's cold at night or because there's no nectar at night. But the outcome of this is introgression, is gene flow. And what you get in these populations is hybrid swarms. Okay? So if there was really nice morphometric differentiation between those two species at the low elevations, that's gone above timberline. Okay? And there's been all kinds of speculation as to why that's true. Elvia Melendez did a beautiful thesis 20 years ago, painting the flowers and asking hummingbirds if they care. And hummingbirds are very interested in the red. They're probably learning it in this case. But also the flare of the nectar tube is important for mechanical isolation because that determined pollen placement on the forehead of the birds. We wanted to do the same thing with hawk moths. We have Hyles lineata in the lab. We had it out in the field in a really good year. And we had a chance with Masha Bischoff to do the same kind of experiment, not only painting them, swapping the colors, but also spiking them with indole. The one major scent difference between the red and white species is that at night, the white flower species makes indole at kind of jasmine concentrations. So indole is easy. You can buy it, you can solve it in acetone, and you can add it to flowers. And so I'm going to show you 3 slides of what happened. When we took both species and put them in an array together without painting them, just spiking the red-flowered one with indole, doubled or tripled the amount of approaches by the moths. That was good enough to get them to approach, but there's no cross-hatching, which means they didn't visit it. Okay? They need to see the flower to poke at it. Okay? And so the white color really does matter. Here, adding indole didn't matter because the flowers already had indole, and there's no dosage effect. Okay? So that was our first hint. When the array is only the red-flowered one, painted white or painted red, there's far fewer visitation by the moths because you don't have an array full of indole, right? So spiking them with a little bit of indole increased a little bit of that attention, but not as much as you got in the previous situation. When you have a mixed species array, when you have the white-flowered one and you paint it red, you reduce a little bit of the visitation. They really like the white color, but adding indole brought that back. And then when you have everybody together with indole everywhere, it's a free-for-all. And this is essentially the hybrid zone. Okay? That's what you get in the hybrid zone. You get everybody in a cloud of indole, and the moths stop differentiating between red and white flowers. And that's really great. It's never been shown before. And what we found was that the attraction of the moths to indole is innate. They come out of the pupa preferring that odor. And we were able to do that in the lab because every moth you see in the field is, of course, experienced. All right, so what have I shown you? Scent can affect seed fitness and structure of floral networks through both attraction and repellence. Scent can mediate reproductive isolation through attraction, preference, and constancy. And the same scent compounds can play different roles depending upon who the receiver is and what the context is. And that's really the rule, not the exception in our field. Perhaps some of the reason why these compounds have diversified and been selected for is that they play so many functions in the life of a plant. And let me just finish by acknowledging the people that I worked with. These 4 different studies were all done in different years with different teams. I never thought as a student that I would branch out like this. I was taught in the 90s that you choose one system and you develop it and then you put your stamp on it and you get your grant renewed. But the world changed, right? And nobody believes in my field, so I have to kind of show them by working with a lot of different people that scent matters everywhere. But it's also enriched my life because I got to work with such different kinds of collaborators in wonderful places. So thanks for having me, and I'd be happy to answer any of your questions.